Okay, now that we've identified a data set that we are interested in finding in the public domain, and we've talked about ArchS4 and the utility it offers us to look for data sets like this one directly within R and Bioconductor, we're ready to see if we can find this data and just pull it really rapidly into our workflow in R. Okay, so I'm going to go over to RStudio now, and you should have downloaded the step 4 script and put it into your working directory. If you haven't done that, go ahead and pause the video and do that now. If I go over to my file browser here and I scroll down, I can see the step 4 script called public data right here. All right, I've opened that script and I'm going to do what I recommend we always do when we start any new script, and that is to go to Edit, Folding, Collapse All. All right, now we can see the code chunks that comprise this entire script, and you remember we can browse through any and navigate to any part of this script over here at the bottom or on the right hand side. So we have our typical introduction and the packages that we'll be loading. We load the ArchS4 database and then interrogate it or query it. We extract data, expression data from ArchS4, filter and normalize that extracted data. Then we go back to ArchS4 and get the metadata. Remember, that's the bit I said is actually harder sometimes than finding the data itself. And now that we have data and sample details or metadata in hand, we go and carry out principal component analysis. Okay? And then we'll visualize our PCA result, and of course all that code is distilled into this essentials code chunk at the end. Okay, so let's get started. We'll skip the intro as usual, and we'll look at the packages that we need to get through today. As I mentioned in the first half of the lecture, there's nothing new here. You should have already loaded the R HDF5 package, which is R's way of dealing with HDF5 files. You already, of course, have the tidyverse. We've talked at length about the packages that comprise the tidyverse. And we used edgeR previously when we needed to normalize our data, and we'll use it again for the same thing in this script. So that's an important thing to recognize, is in going through this exercise on finding and handling public data, it also provides an opportunity to move through some of the things we did in the last script. We're going to need to normalize. We're going to need to create a PCA analysis and then use ggplot to graph that. So we really sort of have this dual purpose today of trying to learn something new, but also getting a refresher on what we did in the last script. Okay, so the first new thing we're going to do here, so I'm going to just, as I usually do, start off by loading these packages, including the tidyverse, R HDF5, and edgeR. And by the way, anytime I've moved to the end of a code chunk, it automatically unfolds the next code chunk. All right. As I mentioned in the first part of the lecture, you should have already downloaded the most recent version of at least the mouse ArchS4 database, and I linked to those on today's lecture page on the data tab of the course website, and I also showed you where you can find this data directly on the ArchS4 website as well. So once you've downloaded it, of course, the first thing you have to do is create a file path that points to it, right? We can't read that data into the R environment if we can't find it. So it's possible you dropped those large HDF5 files directly into your working directory. If so, then your file path will just be the name of the file, okay? I didn't do that. I put these files in my Dropbox folder, 2 directories up from where I'm working. That's the basis for these moving up to parent directories twice, the one level up and then a second level up, and then finding an ArchS4 folder and then finding the data in there. You don't need to do that. I did that simply because that's where I keep the data for you to download, right? It's in my Dropbox in this ArchS4 parent folder. So again, if you just downloaded the data and put it in your working directory, you can just use the name of the file as the file path. Okay, and I have both the human and the mouse. Recall, we only need the mouse for today, but I'm just demonstrating with both. So the first thing I'm going to do is just create a character vector of length 1 that is the file path. Now, when we created file paths before, we used a file path function to do that because we needed to create lots of file paths at the same time from our study design file. We don't need to do that now. We just need a single file path, and file paths are just a string of characters, so we can do that really easily. We put that in quotes and we assign it to an object name. All right, so I'll run these 2 lines of code. Of course, we're not doing anything computationally intensive here. We're just creating this string of characters. And if I evaluate that, I can see indeed it is a character vector of length 1. All right. Okay, now that I have the file paths, I can use those file paths and I can run the h5 ls function, ls here standing for list, and it will look for that file and it will list the contents of the HDF5 file. We won't actually read it or use it or pull anything out. We're just kind of taking a look at what's in that file. So I'll do that for the mouse, not the human right now. And this is our first opportunity to really look at what's inside of an HDF5 file. Now, you remember we talked about HDF5 files being a file system within a file, and that instead of folders and files, we have groups and data sets. And so right away we can appreciate that structure. We have groups and we have data sets. All right. The groups here, I can see there are 3 groups, and they're indicated by this sort of high level designation that the name of the first group is data, and that data contains a single data set within that group, which is called expression. Okay, so data, here it is, contains expression, and that's an HDF5 data set. I can also see the dimensions of that data set, and it's pretty extensive, right? It's 32,544 rows by 284,907 columns. This matrix is really the data that's bundled within this mouse ArchS4 database. It's all the genes by all the samples. So over 250,000 samples, each sample here being a column, excuse me, and each gene being a row. So one of the challenges for today, one of the things we'll have to do is figure out how we slice this large data matrix to get out just the columns that we want. All right, so that's one of the elements within this HDF5 file. Another is a group called info, and I can see that it's a group, and it contains, looks like about 5 data sets. And those data sets are all just really simple. They're strings of length 1, and they contain information about the author, contact, creation date, the lab, version, etc. So we really won't need to access anything within this info group. The third group, and one that is important to us, is a group called meta. And that's where the metadata resides. And it's not necessarily self-explanatory, but it's got all these sample level descriptions, characteristics, contact, the department, the email, the country, raw count data, descriptions, protocols, instrument models, you name it. So we'll discuss this in the script today, but you can see each of these is truly a sample level detail because it's a string with, in this case, characteristics listed for each sample for all 284,907 samples. All right, so we really need to deal with this data group and the meta group. Now, which data set within the meta group is something we have to explore a bit, and we'll talk about that. All right, so 284,907 samples in the mouse database. We won't really deal with the human, but if we just evaluate that as well, we'll see the same exact structure. So you already understand the structure of the human now that we looked at the mouse, but it has 238,522 samples in it. So actually less than what we had in the mouse. Okay, now I've went ahead and denoted those, the sizes of these data matrices within these HDF5 files just for my own use here in the script and for yours. I can tell you these have changed a lot since the last time we did the class. They've gone up a lot, like more data packaged inside these files as they go from version 7 to version 8, which is what we're on now of these ArcGIS4 databases. And we can expect more data in version 9. So now that we understand what's in these HDF5 files, we're going to use the h5 read function. And both h5 list and h5 read, of course, are coming from this R HDF5 R package. Those are functions that come from that package. Now we're actually going to read data from this. And you remember I told you that one of the advantages of working with an HDF5 file is that we don't have to read the entire thing into memory to work with it. We can just, now that we know the structure of it, we can just select the data set we want. So the first thing that I want is actually from the meta group, and it's the sample geo-accession. Okay, so I want to know the gene expression omnibus accession numbers for all 284,907 of the samples in the mouse ArcGIS4 database, because that's what I'm going to use to go and search for just the geo-accessions that are relevant to the study we want to work with today. All right, so I'm going to say I want to read in from this file path just this group and this data set within that group. All right, let's see what we created here. We have something called all samples mouse, and it's a large array. We haven't really dealt with arrays yet, but we talked about them. And it's a character of the length of the number of samples in the database, and it's a one-dimensional array. So just, recall that this is back to what we've discussed earlier with the different types of data types in R, and that what we're talking about is an array, which is, multiple data matrices, but it's only of length 1. So it doesn't really have much dimensionality to it. All right. So we have this all samples mouse. Here it is here. Really all of these geo-accession numbers. And now the next thing I need to do is go in and grab just the locations of my samples, right? So the length of this one-dimensional array is just 283,907. I need to know where my samples are located in that 1D array. So I know my samples, and this gets to that first use case we described in the first part of the lecture of you have a study, you're interested in the samples and in grabbing the samples from that study and analyzing them. So we're going to assume that you know at least the accession numbers from the publication. So here they are, I've listed them, and I'm just creating a character vector of those sample accessions. And for my own use, I've put a comment at the end of each of these lines, and I've stacked the lines to make it easier to spot certain samples and exactly what they are. And I know this because we were involved in this study. We're going to try to infer this information from the metadata bundled within the ArcGIS4. So this tells me the genotype and the treatment and the time point. And I'm going to create this character vector using the concatenate function, the base function to do that. You've used this in your homework already. So when I run this and I take a look at what I created, it's of course just a character vector of length 12. Okay, so 12 samples, and here are their accession numbers. All right, so now I have my accession numbers, and I have this, entire array of accession numbers from the ArcGIS4 mouse database. Now it's a simple base R operation of asking where or which of these samples match my samples. All right, and this operator of percent in percent is maybe more self-explanatory than the Magritte operator. It's a base R operator. It lets us find where in a set of elements where our selected elements reside. So it lets us match or find. Okay, so I'm using the which function, and I'm using this percent in percent function. And And so let's just, probably easiest to just run this and take a look at what we've got. So I'm going to highlight my sample locations, and now I can see that my sample locations is an integer. Okay, so it's a vector, an integer vector of length 12, where each number here represents where in that 1D array from ArchS4 this sample can be found. So this first sample, the one that is the wild type unstimulated rep1, is at location 110,625. Now, it shouldn't be surprising, but just to point out that once I found one of them, I found all of them because they reside in the same location. So samples from the same study are grouped together in the ArchS4 database. So it goes 110,625, 26, 27, and so on to 36. Okay, so that's great. Now I know where they are. So now I'm going to, sort of put that information on the back burner, and I'm going to go ahead and get some more information from the ArchS4 database from that meta group. And this time I want the genes data. All right. So we'll run this. And when I take a look at this genes object, it's another 1D array. Let's see, here it is, genes. One dimensional, it's an array. Where each element in that is the gene symbol. And we know how long this is. It's printed off 1,000 rows, and it's got 31,544 left. So it's 32,544. Okay. We have now the location of our samples. We have all the genes. Now we want to actually get the data. And we need both of these things we created before to do that. We still use the H5 read function. We're still working on our mouse ArchS4 database. But now we're going to go into the data group, and this is the first time we've dipped into that group in the HDF5 file because we want to actually extract the expression data for those 12 samples. We know where they sit, right? We know their column numbers. If we go back to here, we know the location of the data, and we know which rows we want, basically everything, all 32,544 genes, and these columns. So it's another simple operation. We'll use H5 read for this. We'll go into this and we'll ask for a list of length 1 to the length of the genes, all right, for my sample locations. So we're asking for really all of the genes, and this keeps us from having to specify the exact number. We just say we want 1 to the length of this object that we pulled out here, and we want the data for those rows for just these sample locations. All right, so we'll hit run and we'll take a look at what we've generated. Looks like we got some data. It's a large matrix, okay? And if we actually open this in our file browser here, we can see we don't have column names. That's okay. We don't have row names even. It's a data matrix. That's okay. But we can see we have counts, and we have the right dimensions here. It's 12 by 32,544. Great. So now that we have this basic element, we've really succeeded in what we set out to do, right? We reached into this really large database packaged inside an HDF5 file, and we pulled out just the data corresponding to our query. So if you have geo-accession identifiers or other identifiers, you could do this easily for any samples you wanted to find. And as this ArchS4 or the DEE2 that I showed you in the last part of the lecture, or other databases like it, continue to expand and grow, the likelihood that you would be able to find a data set in there, I think, is only going to get better, right? Because they're including more and more data. All right, so I do want column headers and I do want row names on this data, of course. So now I can take that genes element I pulled out and I can just assign it to the row names of this data. And this isn't using tidyverse or anything like that. We're just using basic functions here in R. So I want to assign this genes object to the row names of the expression data. And remember, this is a data matrix, so I really can only put this kind of information in the row names slot. All right, now if I take a look at expression data, now I've got all the row names here. We also want to get the column headers on, and I know I have these column headers because this object all samples mouse, which is all of these geo identifiers, and my sample locations, right, just where mine are, I can now use that as a subsetting operation and say, listen, I want to take this 1D array and I just want to return the elements from it that match these numbers. All right, and I want to put those into the column names component of my data matrix. All right, we run this, we'll take a look at what we have now. I think we need to update this. Let's just open that up. And there we go, right? So now we've got all of our gene symbols and all of our accession IDs. Okay, so now we can see if this makes any sense at all, right? Do we have data that looks like sequencing data? So I showed you this before when we were looking at our own data, but let's run the call sums operation on our expression data. It'll tally up each column, and we understand, since we're dealing with counts at this point, that that should be the sequencing depth for each of the samples we extracted. And sure enough, these numbers look, what we might expect for mammalian samples or tissues. They're on upwards of 20 to about 38, 36 million, with a couple samples that are lower. Here's one that's about 19 million, etc. Great. So now that we have that, we're ready to move forward with exactly sort of what we had done in previous steps of our scripts, like using EdgeR's DGE list function to turn this expression data into a digital gene expression list. Very easy to do. And when we do that and we take a look at what we've got, it really just creates a list of length 2, where the first element in the list is the data I just showed you, a count matrix, and the second element in the list is sample level information with the name of the sample and the library size, and no normalization factors yet, but we'll get there. Okay. Now that we have this DGE list, we can easily calculate our counts per million. And when we do that and tally that up using call sums, because by its very nature we had converted into counts per million, each column should total 1 million, and it does. Great. So we've really achieved the first main part of our goal, pulling out all the expression data only for the samples we want. So now we want to do the things we had done before with actually normalizing our data, filtering it, normalizing it, and then representing it as counts per million. So I'm going to take you through the steps. We already did this with our own data, but in this case, I'm asking, how many samples, right? How many rows, how many samples have zero, how many genes have 0 counts in all 12 samples? All right, I'm just curious. I'm not filtering yet. I just want to see a table of how many genes have no counts, no reads mapping to them in all 12 samples. And I can see that at least a third of the data, or about a third of the data, is true for that operation, meaning they have 0 counts in all 12 samples. They're simply not expressed. So clearly there's filtering that needs to be done. I'm not going to filter that stringently because I don't want to lose samples that might have 0 in a couple samples but not in others. So I'm going to use the row sums function operating on my counts per million object that I generated up here, and I'm going to ask to only keep samples that had greater than 1 count per million in greater than or equal to 2 samples. And I choose 2 here because I'm working with duplicate replicate samples. All right. And so that should create an object I'll call keepers that I can then use to subset my DGE list. Let's take a look at what keepers looks like. All right, so it's a true false, obviously a logical structure here, and it tells me true or false whether each of these gene symbols actually met those criteria. So now I can use that to filter just the rows, right? So this is a subsetting operation. I'm performing or operating on the rows because I'm to the left of that comma, and I'm not subsetting the columns. There's nothing to the right of that comma. And this is all going to subset the thing that's in front of these brackets. All right, so we'll run this and take a look at the dimensions of what we produced. And now I see that I'm down from 34,000, whatever it was, to 12,000, right? So major attrition here, which is fine. And we can, debate about what's an appropriate sort of filtering stringency. And there's no right answer for that, but for the purposes of this class, I'm just demonstrating that we, A, need to filter and that we can easily decide on those criteria and apply them. All right, now that we've filtered our data, we'll normalize, and we'll do exactly what we did before. We'll use the calc norm factors and the trimmed mean of m values method that's available to us through edgeR, and we'll operate on that filtered data and produce a filtered normalized data. And so now, when we take a look at this new DGE list, now we still have our counts data as the first element in the list, and our samples, we now see that we have normalization factors here. Great. So let's now extract our counts per million in a log2 scale from this normalized filtered data, and we'll store that in a data matrix called norm log2 counts per million, which looks like this. Okay, so that's it. We filtered and normalized our data, but we still have yet to get metadata from that ArchS4 database. So although I know what these samples are, we don't have any way to really attach meaning to them outside of what I'm telling you here. So if we're treating this as an exercise where you're going to be pulling data from the public domain and you need that metadata, then we need to go through how to get that. All right, so I'll just reiterate again, I find this to be one of the most challenging parts of handling public data. There are structured languages or ontologies for how we annotate or how samples and how we describe samples, the language we should use, but we rarely in biology adhere to ontology standards, which means, metadata is often unstructured, unfortunately, and again, the rigor with which authors and data repositories mandate the extent and clarity of the metadata is really lacking. Okay, so what do we do then? Well, we're going to still go back to our ArchS4 database. We're going to use that the HDF5 H5 read function working in mouse, and we're going to dip into that meta group again, but this time we're pulling out the sample source name. Now I'm going to just quickly go back up here because I want to just list what's in this HDF5 file again so that we can see we're really operating in this meta group, and there's a whole bunch of stuff here. How did I know to go get this one data set from that group? And the answer is I just guessed, and it was a little bit of a look and see operation where I dipped into each of these groups and peeked at what was present in those data sets. I think it's safe to say we don't need contact name, contact phone. So some of these I was able to ignore because they did a good job naming them. And sample characteristics is another term for metadata. Okay. So what I don't know what CH1 means here, but this was a logical place to look, and indeed there is valuable information there. But not just there, there's also information that's useful to us in other parts of this, like a sample description, a sample title. So what we're ultimately going to do in this part of the script is go through and grab several of these data sets from the meta group. So we're right here. I'm going to go in and grab this sample source information and sample characteristics and sample title. All right, and I'm going to store each of those in an object that just copies that name, really. All right, so I'll run each of these, and I'll highlight one so you can take a look that what we're dealing with here is, again, a one-dimensional array of the length of the database. And if you look at the sample source Here's what that looks like. We can see it looks like tissue or cell type. If we look at sample title, that should be really effectively a sample name, okay? And if we look at sample characteristics, that's probably going to be a more complex field than it is, with sort of colons separated, it looks like, various information about strain, about tissue, about sex. So that's really, I think, between sample source, name, the sample title, and the sample characteristics, I think we should be able to piece together what was done with these 12 samples. So now that I have these, this information pulled out of the HDF5, I'm going to use the tibble function to just make a tibble from each of these, to combine these as a tibble. Obviously, I don't want the whole list, right? They're 284,000 long. I want to just take the information from these objects for my samples. So I'll use that my samples location to subset these 1D arrays. And I'm going to give each of these a title name, a column name, sample title, source, and characteristics. That will create the structure of my tibble. And since effectively what I'm trying to do here is create a study design analogous to what we have for our own data in the course, I'm going to assign this to an object called study design. All right, let's take a look at what we've created. It's a data frame, of course, a tibble, 12 observations of 3 variables, and this is what I have. I have sample title, sample source, and sample characteristics. So if we take a look at this right away, I can see that, in our case, I can pat ourselves on the back a little bit. We did a good job naming our samples in a way that we could get a lot of this other information just from the name. The genotype, how it was treated, and the replicate number are all encoded in the sample name. I also have that additional information here as well, so if it wasn't apparent in the name. And I have more information over here as well. So it looks like I didn't need all 3 of those columns, but now that I have this, I need to just clean it up a little bit. So great. So if we scroll down now that we've created this tibble, now I know from looking at the study design I created that I can just now clean this up and structure it in a more clear way. I still want a column called sample title, but now I see that there's really 2 variables here. There's genotype and treatment, right? That's evident here. There's a genotype, there's a treatment, and there's replicate number. So I'm going to go ahead and just set up a 3-column study design, making a tibble with the first column being my sample title, the second column being genotype, and the third column being treatment. And I'm just going to create these character vectors myself from what I inferred just from looking at this. If I run this and now take a look at study design, now I have something that looks exactly like what I want for a study design, right? Clear sample titles, a variable called genotype, a variable called treatment. Those are the 2 variables I'm going to want to explore. Okay, great. Now that I know those are the variables I want, let's go ahead and capture those variables from the study design, turn them into a factor, right? We've talked about this in the last lecture. And I also capture the sample title and assign it to an object called sample name because we like to use that to throw on graphs. So now we have genotype, we have treatment, and we have sample name, right? 3 different character vectors. Okay, now that we have our data and we have our study design, we're ready to carry out a principal component analysis. Exactly as we did in the last class in the sort of multivariate analysis part of the course, we're going to use the PR comp function to run our PCA. We're going to take a look at the summary of that. We're going to pull out this X element that contains information about how much each sample contributed to each principal component. We're going to convert our eigenvalues into actual percent variance, and then we're going to visualize that PCA. So I'm going to move through this quickly because we've done this before. And I'm going to just look at this last bit, which shows me that there is a lot of variance in my data that can be accounted for by PC1. Vast majority of the variance in gene expression in this set of 12 samples is accounted for by a single principal component. So obviously I'm really going to want to know or hopefully assign some biological identity to that principal component. Okay, now that I've generated the principal component analysis, I want to plot it. So we can go ahead and do that just as we did before using ggplot. What we need, of course, to turn it into a tibble first, the PCA res $x. And then I'll plot it, and we'll take a look at what we've got and talk about how to improve it. Okay, let's go over to the view, plots here and see what we've got. Oh, I didn't run it yet, sorry. There we go. Okay, so here's our PCA. Remember, I have my coordinates fixed, so it actually appropriately scales this x-axis to represent the fact that this is really where most of my variance is. If I turned off that coord fixed, and we talked about this in the last lecture, and I reran that, I would end up with a PCA that just doesn't accurately reflect. If my window was even smaller, it would be even worse. Doesn't really respect the ratio between these two. All right, so important to recall that that's an element of our PCA that we want to make sure is represented in the data, in the plot. Okay, I can see, sure enough, PC1's really driving apart these samples, and to a much less extent, PC2. So I'd like to paint information onto this, right, that we pulled out of the ArchS4. So why don't we take a look at, for example, mapping treatment to color, right? So I'm going to say color equals treatment, all right? And treatment here was stimulation with LPS, a really potent microbial component. And sure enough, LPS is driving a huge amount of the variation in the entire data set. That's not surprising if you're an immunologist or somebody who's worked with LPS and understands how potent of an innate immune stimulus it is. That's been well known for a while, and we certainly don't want to report what other people know very well, that LPS is a potent stimulus. So this data set's an example of how the first principal component, although reassuring to see that PC1 is LPS stimulation, it's really not of interest. It's going to be subsequent principal components that make up a far less percent of the variance that are of interest to us. So one of those other variables we mentioned was genotype. And so we're going to set that to shape and rerun this. And now what we can see here is that PC2 really does separate our genotypes. We see that these double knockouts that lack RIPK3 and caspase-8, important components of cell death pathways in the inflammasome, are really responsible for this substantial but modest compared to LPS percent of variation that separates from the RIPK3 single knockout and the wild type. So this tiny amount of variation, 2.9%, is actually the basis for this entire experiment and for the paper. All right, so we could continue to explore this PCA, and one of the ways I described doing that in the last lecture was actually taking this data and creating a small multiples plot. And so I'll take my PCA res $x, and I'll take just the first 4 columns of that principal component analysis, so PC1 through 4, and I'm going to use that to make a tibble where I've added a column for the treatment variable, the genotype, and the sample name. If we run this and then take a look at what we've got, this should be pretty clear that we've created this, okay? Once we've created that, we can now use the pivot longer to take our messy data where we have one variable, principal component, spread across multiple columns, and we can take those and put them in a single variable column like we have for treatment and genotype, and take all the data and put it in a single column for the loadings. So we do that, and we take a look at that pivoted data. And now this is very easy to plot and facet by the principal component and pull out all the numeric data as a single numeric vector. So we'll return to ggplot for that, and I'll paint the fill of these bars. I'm going to use the geometric bar as treatment, and I'll coordinate flip so that our sample labels, which are kind of long, will fit on there easily. And this may take just a second to plot, but there it is, right? So these sample names are now easy to read because I'm not trying to look at them standing up vertically. And just as I saw in the last plot, LPS treatment with LPS is really driving this first principal component. Any sample treated with with LPS is weighing on PC1 in one direction, and anything that's unstimulated is weighing on PC1 in the other direction. All right. And that treatment doesn't really correspond to any of the other three principal components, 2, 3, or 4. So what about the other variable, genotype? Remember, we already saw that that accounts for PC2. Is there more there than just PC2? So we'll paint on a different color onto this small multiples plot. And we have 3 genotypes, so it gives us 3 colors. No relationship to PC1, but lo and behold, there's that PC2 separating RIPK3, which are weighing on PC2 in one direction, with RIPK3 and wild type in red and blue, respectively, sort of weighing more modestly, but in the other direction on PC2. But what I can see is that PC4 also looks to have some relationship to genotype, because all of wild type are going one way, and almost all of the other genotypes, the knockouts, except this one sample, are weighing in the other direction. That suggests to me that if I really want to create a PCA that gives me maximal resolution between my genotypes, I might want to plot PC2 versus PC4. So I can go back up and actually do that, make X PC2 and this PC4, and just do color as genotype. And this, of course, now needs to be labeled as PC2 and PC4, and needs to appropriately draw on the correct percent from my object there. All right, so let's see what we get when we run this. Great. So perfect, right? Now I can see clear separation between genotype. And since that's really the subject of this story, even though these percent variances are really very low, we know that LPS is a real sledgehammer, and now we can dig below that sledgehammer effect and start to get at what we really care about. Okay, so that really brings us to the end. The rest is just everything I showed you distilled to the essentials. So hopefully now you understand how to pull out as much data as you want, along with the metadata from the ArchS4 database, whether you're dealing with human or mouse. If we just then take stock of where we are now, we can either use SRA tools or GrabSeeks, which operates using SRA tools to get access to raw FASTQ files. Or if we use different aligners like Bowtie2 or HiSat2, we can use their sort of SRA aware alignment function to directly align reads using just the accession numbers. But we also understand that we have available to us these projects like ArchS4 and like DEE2 that we can tap into at the command line and pull in data, really bypassing the need to hold these big FASTQ files and working directly with Callisto aligned data that we're already familiar with, that we can move through our pipeline and be at visualization very quickly. Great, that's it.